Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Evaluating the Impact of Cell Culture Supplements of CHO Clonal Outgrowth Efficiency. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. So just type those questions in the chat box by selecting the questions icon located on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the Comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Nova Biomedical, who developed the content for this presentation. Advanced Instruments and Nova Biomedical are now doing business under the same brand, bringing together decades of expertise in analytical instrumentation, R&D, and global customer support. The Solentum ecosystem is now part of Nova Biomedical's portfolio, providing best-in-class imaging and single-cell disposition technologies for cell line development, workflows, and assurance of clonality for regulatory bodies. We had a change in our speakers today. Unfortunately, Adam cannot be with us today, but we do have, replacing him, Noud Drumming, Field Application Scientist with Nova Biomedical, and there he is. Now partners with cell line development teams across Europe, the Middle East, and Africa to optimize workflows, streamline processes, and drive innovation in laboratory operations with a strong focus on Solentum solutions. He combines his strong scientific foundation with deep knowledge of the life sciences market. Whoops, I forgot to put on my webcam to welcome him. He combines his strong scientific foundation with deep knowledge of the life sciences market to help laboratories enhance efficiency and accuracy in single-cell seeding, culture monitoring, and confident clone selection. So thank you very much, Noud, for joining us today. Next, I'd like to welcome Mohammed Al-Badr. He is a business development and marketing manager at UGA Biopharma, where he focuses on promoting and supplying advanced CLD services, new biological entities, and biosimilars. He plays a key role in supporting UGA's portfolio, including the R2U cell lines designed for blockbuster biosimilar production, as well CHO Prime Media and Feed platform. Through his academic journey, Mohammed developed strong hands-on expertise in mammalian cell culture, molecule cloning, and recombinant protein expression. He combines this technical background with a strategic business perspective to drive growth and innovation in the biopharmaceutical sector. And next, I'd like to welcome Dorothy Dusterhoff. She is an associate scientist in the cell line development at the UGA Biopharma, where she develops cell lines for therapeutic drug candidates used in commercial manufacturing. Her work focuses on optimizing single-cell cloning through a systematic evaluation of cloning supplements, implementing high-throughput approaches, and applying quality and regulatory principles for monoclonality assessments. She also designs data-driven experiments to accelerate CLD timelines. So welcome there, Dorothy. Now, with the introductions out of the way, it is now my pleasure to pass the mic over to our first speaker. So now you may begin.
Good. Thank you, Sonia, for the introduction and for hosting the webinar today. And a special word of thanks also to Mohammed and Dorothy, who have agreed to do the webinar today. We've been cooperating with them, testing our cell culture supplements, and they'll provide lots of the content today. So thanks again for cooperating with us. I'd like to start with providing an overview of what we're going to cover today. So we'll start with an introduction into the Solentum reagents. Then Mohammed and Dorothy will take over for the main part of this webinar today with their UGA Biopharma case studies on the InstaGrow CHO Plus and the InstaGrow CHO CD reagents. And we'll wrap it up by talking about the benefits of chemically defined reagents in cell line development workflows. Hopefully today all of the attendees will gain actionable insights of how to maximize CHO cloning efficiency. But before we start, we have a poll question we'd like you to answer. Sonia, if you could pop up the question, please, for us.
I will certainly do that right now, and there it is, and everyone should see it appear on their screen. And there we go. So which field best describes your area of expertise? Now, I'll give you 45 seconds to complete this simple question. This is done in real time, so your participation is strongly encouraged and very much appreciated. So here are the options. Which field best describes your area of expertise? Is it CDMO, CRO, biopharma, academic research, or regulatory authority? So please go ahead, everyone, now and cast your votes. Okay, that looks like it's almost half of our audience participating. So that's very greatly appreciated. Now, if I can just encourage everyone else to jump in there, please. Fantastic. Okay, so that 45 seconds is up. Thank you, everyone, for participating. Now let's take a look at who we have in our audience. Okay.
All right, thanks. Sorry.
Oh, there you go. Do you see the results there?
Yeah, I've seen it on my screen at least.
Yeah, so CDMO, we got about 18%, biopharma, 64% of our audience, academic research is 18%, and regulatory authority, 0%. So thank you very much, everyone, for participating. I'm going to close the polls, and back to you now, Naud.
Thanks, Sonia. All right, so yeah, let's start with the introduction to the Solentum reagents by Nova Biomedical. As a high-level overview, and I'm sure you're all very aware of how cell line development works, but I want to start with providing a bit of overview of some of the challenges that we're trying to tackle here. So cell line development is a critical process within biologics development with the goal of generating a stable clonal cell line that can produce large quantities of good quality biologic, and that can either be the cell line product, so for example, a monoclonal antibody, or the cell line themselves, for example, stem cells. So typically, for this overview, we'll start at the transfection stage where we have millions of cells per milliliter, which have been transfected with the gene of interest. And then at some point during cell line development, we will perform single cell isolation to ensure monoclonality. All those single cells will then be monitored for outgrowth, and we'll select the best performing ones on multiple criteria to be passed on to next steps. And this is what we call clone selection. So we're trying to find the best performing candidates, the most promising clones in our pool. At the end, we'll do master cell banking, where we're going to cryopreserve these high potential candidates for further testing, for example, for scalability or consistency. And unfortunately, this comes with a number of challenges. So at the very beginning, when we're reviving our host cells, we'll have slow recovery and low viability of these cryopreserved cultures, which reduces efficiency and extends timelines. After single cell isolation, we can see poor clonal outgrowth, imaging artifacts based on the media or supplements that you're using. Both will increase our hands-on time and therefore cost, and will also reduce our regulatory certainty. If we're performing scale-up, for example, we're transitioning from our static to shaken cultivations, we lose clones during this process due to poor adaptation, and when we're doing our master cell banking in the end to cryopreserve our highest potential clones, we can again see low recovery post-cell banking, which again adds uncertainty to our workflow. The Solentum reagents are specifically designed to overcome some of these challenges, and I'd like to give you a brief overview of the different solutions that we have for each of these stages within cell line development. So first of all, and the main topic of today's webinar is InstiGrow CHO and InstiGrow CHO CD, which stands for chemically defined. So these are to enhance growth and cloning efficiency of suspension CHO cell line development workflows with animal component free, and for the latter, chemically defined supplements. We also provide in CHO, which maximizes viability and viable cell density during clonal expansion, moving from static to dynamic cultures within CHO workflows, as well as in-thaw CHO, which helps to protect your cells during cryopreservation. All the reagents that we have here are animal component free. They're specifically designed for cell line development of biologic drugs, and therefore animal component free for regulatory compliance. They're easy to use. They're simply diluted directly into the cell culture media to working concentration, in the example of the in-situ CHO, to improve clonal outgrowth. The industry proven, we have numerous world-leading service providers, pharmaceutical and biopharmaceutical companies working with our reagents in stable CHO cell line generation workflows. And the newest addition to this portfolio, the chemically defined in-situ CHO, has been launched due to a growing demand for regulatory compliance and reduced variability between batches. So if we look at these, the in-situ CHO reagent, there's a couple of benefits that it was purposely designed for. So first of all, it's very easy to use. As I mentioned in the previous slide, it's simply added to the cloning media before single cell seeding, and it has an unmatched optical clarity. This is purposefully designed because at the early stage imaging, it's essential to get the highest clarity possible to get the images that you need for your IND submissions without any uncertainties about debris or particles in those images. Furthermore, they are designed to enhance clonal outgrowth. We have some data on the right-hand side that shows how the reagents provide crucial components that generate more clones with improved outgrowth during single cell seeding. So you can see the top image here shows a plate without in-situ, so this is just a conditioned medium, and the lower image shows a plate that was seeded with in-situ. And we can clearly see much higher outgrowth rates and much higher confluence levels achieved in the place where the in-situ was added before seeding. With that, I'd like to hand it over to UJ Biopharma, to Mohammed and Dorothea, which will tell us a bit more about how they use the reagents in their workflow. Mohammed, please go ahead.
Thank you, Noud. So, good day, everyone. Allow me to start introducing myself. My name is Mohammed Al-Badr. I am the Business Development and Marketing Manager at UGA Biopharma. I will walk you through a few slides introducing the company, UGA Biopharma, what do we do, and who we are. Going on. So, UGA Biopharma is a German CRO, a contract research organization that was founded in 2009. And it is located in a small city on the northern borders of Berlin called Hennigsdorf. We are, as a company, we are a mid-sized company. We are 45 employees, and we are proudly very happy. We are proud that we are certified by the ISO 9001 regarding to the QMS, the quality management system. Through the years, we have successfully finalized more than 50 projects to develop cell lines for producing of novel biologics in addition, as well as biosimilars. Moreover, we have already developed more than 20 RTU cell lines, and I will talk about this a little bit more to details in the coming slide. Moreover, we have provided our clients with more than 4.5 million liters of our proprietary media and feeds products. The UGA Biopharma portfolio consists of two main parts: the R&D services, in addition to the catalog products. What is our R&D service? What we offer under R&D services? We do offer cell line development, where we, through the cell line development, starts usually for the DNA material or sometimes even DNA sequence. We go through the steps of this cell line development, starting from pool generation up to clone generation and clone selection. The anticipated productivity of these cell lines increases from around 1 gram at the pool generation phase up to 8 grams by the in-phase the clone selection. Moreover, we do offer development of an upstream processing where we in parallel screen several parameters for the process in a small controlled cultivation system, such as Ampere 15. Moreover, we do process optimization in 1 liter bioreactors, and we scale up in the R&D scale up to 5 liters bioreactors. And last not least, of course, we do the tech transfer of the process to the client's facilities. Moreover, we do develop the downstreaming and analytics methods for our clients and for the processes, including a very long list of analysis that is very helpful for the clients and the approval of such products if we're talking about a novel biologic or a biosimilar, including the very well-known the CV analysis, the charge variants, in addition to the Anglicam profiles. And this will help assess the product, especially if we're talking about a biosimilar, the similarity of this biosimilar to its originator. Moving on, the catalog products have 2 streams at UGA Biopharma. The first stream is the RTU cell lines, which I've already mentioned. The RTU cell lines are cell banks which are on the shelf ready to be used to produce your own biosimilar for a client that you would like, for example, blockbuster biosimilar such as Humira, the adalimumab, or another example would be Keytruda, pembrolizumab, where these are cell banks on the shelf, could be acquired and start developing your own biosimilar. In addition, we have specific proteins that could be also as on the shelf that could be also acquired, such as the bispecific antibodies, chimeric proteins, enzymes, in addition as well as hormones. On the other hand, we do offer as a catalog products our own proprietary feeds, media and feeds platform, which is called CHO Prime. It is optimized for CHO cells to ensure higher productivity. Meanwhile, superior product critical quality attributes, and this could be ensured or reached through minimizing the stress metabolite formation. The media and feed platform is chemically defined, is animal and human free component, so it's free of these components of animal and humans, and the compatibility between the different SKUs is no problem. You can easily mix these, and this could be very helpful. For instance, the right mix ratio between a CHO Prime standard with a CHO Prime LG, and in this case, the LG means here the low galactosylation. This will ensure to be able to adjust your product or the intended biosimilar to have the perfect Anglicam profile and to assess the best similarity with its originator. So I would like now to hand over to my colleague, the star of this event, of today's event, Dorothy.
Thank you, Mohammed. I would then like to start with the case study. First of all, my name is Dorothy. I'm an associate scientist at Cell Line Development at UGA Biopharma, and I would like to give you first a short overview about our cell line development process that we have at UGA Biopharma. We start first with the vector design and construction. We used our patented in-house vector platform to transfect our in-house CHO cell line to generate stable pools, which then express our molecule of interest. After transfection and pool generation, we expand our pools and screen them for productivity. And from the most promising pools, stable clones are then generated. This is done by single cell cloning, and from the clones that outgrow, clones that get expanded and then screened by a clone productivity test. We are looking for the most promising lead clones that express the product with high yield, show good growth attributes, have satisfactory quality, and of course a stable expression. As I mentioned, for the clone generation, one part of it is the single cell cloning. The single cell cloning is mandatory as cells that are produced for therapeutic usage, as for expressing monoclonal antibody, must be proven of monoclonal origin. This is done to ensure that the later product, ensure quality, consistency, and traceability. There are different methods how to generate single-cell clones. Originally, it has been done with limited dilution, but more prominent now are single-cell sealing devices, as the VIPs, which is part from the Solventum portfolio that we have in-house. Our proof of monoclonality at UGA is done by an image-based proof of monoclonality, and for that we use the cell-metric imager, which is also part of the Solventum portfolio. And we use it to generate whole whale image, which then follows the proliferation from a single cell into a colony. On this slide, you can see clone report that can be generated with the cell-metric software. And what you can nicely see is that on day 0, there is one cell present, which then proliferates into a colony after 14 days of cultivation. However, one important question to address is how to actually achieve clonal outgrowth. There are different supplements that you can add to promote cell growth. The most widely used one is the fetal bovine serum, which has been widely used in vitro cell cultures. However, when you work with cells that have a therapeutic usage, it's not recommended by regulatory authorities, and it is of animal origin, which can cause risk of contaminations, as well it has also risk of batch variations and not a defined composition. One other supplement is conditioned media. Conditioned media refers to media where cells which has been used to grow cells in it, and these cells have secreted growth factors into the culture. And by filtering it and mixing it with fresh media, you can use it to promote cell growth. However, also here, there's a high risk of contamination and it has not a defined composition. And furthermore, it's not working for every cell line. So for us at UGA Biopharma, the only option is the serum-free media supplement. And when we were looking for a reliable growth supplement, we are looking for one that is animal component-free, has a defined composition, significantly increases cloning efficiency, has low batch variations, and as we work with an image-based proof of monoclonality, the supplement must preserve optical clarity. And since it's also a very important product or reagent in our workflow, we need a stable supply chain. In 2023, we started our initial experiments with InstiCHO Plus. InstiCHO Plus was very interesting for us because it's animal component free and it claims to keep optical clarity after filtering of the product. In our initial experiments, let me shortly make this a pointer. In our initial experiments, we compared a control supplement, which was a commercially available supplement, against InstiCHO Plus. And what you can see is that for pool 1 and pool 2, the control failed to work. while with the first results we got with the InSeq Plus were quite promising, as we saw clonal outgrowth after 14 days of cultivation. But before I will go into detail on our initial experiments, I would like to generally show you our experimental setup. This will be quite similar for all the experiments that will come. So what I did was that I started with the media preparation. There it was important to filter the InSeq Plus prior to use, and then I added it to the cloning media. For the limited dilution, I used mini pools from a CHO cell line that expressed monoclonal antibody, and I calculated a seeding density up to 0.75 cells per well in a 96 well plate. After the limited dilution, the cells were cultivated under static conditions for 14 days, and then a whole image was taken. This was done by the Cymetric imager, and this device not only imaged the cells, which you can see here, it has also a software feature which you can use to calculate your cell confluence in a well. And for instance, then the software recognizes your colony and then marks it green. So for this culture or clone, a cell confluence of 15% was calculated. In my initial experiments, I seeded 2 mini pools with a limited dilution. I tested a control supplement against InSeq Plus, and I have here 2 diagrams. So I have, for once, the average clonal confluence on day 14. This gives you an impression on how big the clones were that grew out after 14 days after cultivation. And on the right side, I have a diagram for the cloning efficiency. I would like to define what we understand under cloning efficiency, as it gives us the number of clonal colonies divided by the single cells seeded in total. And we do only take colonies into account that have a clonal outgrowth above 5% on the last day of cultivation. So this cloning efficiency gives you an impression about the quantity or the number of clones generated. And what you could also already see in the whole image was that the control failed to give clonal outgrowth at all for pool 1 and pool 2, but the InSeq Plus showed really nice results, so we got a high number of clones, which was quite promising. And to implement it into our workflow, we wanted to test the ideal working concentration. For that, I tested 1 mini pool and different InSeq Plus concentrations in the limited dilution. And the recommended working concentration by Nova Biomedical was 1 time InSeq Plus, but we saw that we got the best results with 2 times InSeq, as it is the more suited concentration for our in-house cell line and our in-house conditions. To test the batch-to-batch comparisons, I tested 3 different lots of InSeq Plus in a limited dilution with our now with 2 times InSeq Plus, which has been shown to be our most suited working concentration. The left 3 bar graphs are always showing the 3 lots for pool 1, while the right bar graphs are showing the 3 lots for pool 2. And why we got an overall good cloning efficiency, so with all 3 lots we saw clonal outgrowth and also good clonal outgrowth around 10 to 20%, we saw that there was still some batch-to-batch differences, as the second lot showed a weaker performance in comparison to lot 1 and lot 3. With this supplement, we finished some of our projects and we're really happy about it. And however, we were really interested when in 2024, Advanced Instruments, Nova Biomedical asked us if we want to test the new version of CHO Plus. And based on the results from this case study, we published an application note together. The experimental setup was that I seeded 2 mini pools with a limited dilution, and I tested the original CHO Plus version against the new CHO Plus version, and also took a control into account. For every condition, I tested 3 96 well plate with our UGA media and 1 96 well plate with the commercial available media. On this slide, you can see the results from this case study, and on the left side there is the cloning efficiencies shown for pool 1 and pool 2, and on the right side there's the average confluence of clones for pool 1 and pool 2. And the most prominent color that will pop into your eye is, for all graphs, blue and orange. The blue graphs are showing the new CHO Plus media, CHO Plus variant with our UGA media, and the orange bar graphs are showing the original CHO Plus variant with our UGA media. And we are really happy about these results, as we can see that, or we were happy to observe that with the new variant, an increased cloning efficiency was possible. So it was able to improve the cloning efficiency from around 10% for both pools to up to 30%. And also, yeah, we were seeing that it has a benefit on the average confluence of the clones. So we get more clones and bigger clones with this supplement. We are really interested in implementing this new version into our workflow, but first we tested again the most suited working concentration. And I did this again with a limited dilution, testing 2 mini pools and testing different integral concentration of the new version. And what stood out is that one time CHO Plus was the most suited working concentration. As you remember, with the older version or original version, we needed 2 times CHO Plus. So with the new supplement, we saw an increased performance, but we needed less of the supplement, which saves us money and costs. And yeah, we were really happy with this product. Here I want to show you some of our project that we finished with, or that we successfully finished with the new CHO Plus version. And what I want to point out is that we had one project where we had like really high cloning efficiencies, up to 60%. And what does visually mean, you can see in the whole image. So there are many clones to screen. And for this project, we saw that all pools that we seeded had a really high cloning efficiency, which is nice because we get more clones to screen and a higher, maybe a higher chance of finding a really good lead clone. We do not only see that we get many clones to screen or a high clonal outgrowth. We also get clones with a high clone productivity test, which you can see here in this graph, showing that the clones that we screen also are showing high clone productivity titers, and we were able for this project to reach a product titer of 6.5 grams per liter after process optimization. However, I want to also show you another effect that we saw. For some projects, we see or observe pool-to-pool variations, meaning that for pool 1, which is shown here in this bar graph, we got high cloning efficiency, so many clones to screen. But for pool 3, which is shown here, we got this result after 14 days of cultivation. So we got a few or a very low cloning efficiency, meaning a very low number of clones that we could possibly screen and expand. And for the whole project, there was a big heterogeneity and big pool-to-pool variations. And this is undesired, as for this project, the pool 3, so this pool, was our most promising one. So the one that showed in the pool screening the highest pool titers. So we expected to have also the most interesting clone candidates from this pool. And what we had to do was to repeat the single cell cloning for this pool with a higher amount of plates, which costs us money and expanded the timeline. So even though for some projects we saw really good results with the InSeq Plus version, for some we still observed difficulties as pool-to-pool variations, and still we also saw some batch-to-batch differences when ordering a new lot of InSeq Plus. Based on these difficulties, we were really interested when Nova Biomedical told us that they will bring a new product on the market, which is a chemically defined version. And we, based on the results of the case study we did, we published a second application note, which got online, I think, in beginning 2026. So, and what I did in this case study was again testing too many pools with limited dilution with our new UGA media CHO prime, testing 2 lots of the InSeq CHO Plus version against the chemically defined version InSeq CHO CDX. And on this slide you can see our first results. So what stood really out was that for every supplement we used, we got an equal number of clones. Nevertheless, we used InSeq CHO Plus or InSeq CDX. However, what was really impressive was that with the InSeq CHO CDX version, the pool-to-pool variabilities could be decreased a lot. So we have lower pool-to-pool variabilities in comparison to the InSeq CHO Plus versions. With this variant, it seems that pool-to-pool variations could be prevented. However, there's still the question on if also with a chemically defined version, the batch-to-batch differences could be overcome. And this is something I want to test in further experience. So I would like to give you a short outlook on what we are planning to do. We want to test InSeq CDX with different molecules and host lines and test different batches for batch-to-batch comparison, and also want to test compatibility with our CHO prime AG media, which is optimized to control N-glycan profiles. With that, I will come to my end of my case study, and I want to point out that, since our collaboration with Nova Biomedical beginning in 2023, we saw that there was a constant improvement of the supplements, and optical clarity of the product was always ensured. We saw compatibility with our CHO prime media, and we're happy to successfully complete many customer and in-house projects with the InSeq CHO Plus supplements. We really appreciate the close cooperation and the good communication and furthermore, the fast delivery of the products. With that, thank you for your attention, and I would like to give the word back to Naut.
Thank you, Dorothy, and thank you, Mohammed, for the first part. Yeah, great to see the results from your case studies, and it's always good to see customers using our. Yeah, thanks again for that. I'm just going to wrap it up with a quick overview of the benefits of using chemically defined reagents. Dorothy already mentioned they've tested them. Let me just show again. Yeah, that they've tested them in their workflow, and I just want to talk a little more about the benefits of using them. So, first of all, a chemically defined product is chemically defined if we know its exact composition. So it cannot contain any raw materials with a complex structure or unknown composition in general. We've gathered feedback from our customers, and we'd like to share some of their perspectives. So, for example, from a service provider perspective, we had the feedback that their team was impressed with the Outgrowth supplements, but that they did have a concern around the fact that it isn't chemically defined, because sometimes their clients will request or require it, and they want to be comfortable in knowing that they have chemically defined reagents in their toolkit before engaging with their clients. From a developer perspective, we have heard the feedback that they need to provide evidence that the materials that they use are chemically defined to regulatory instances. And we have basically pulled all the feedback that we got and saw two main advantages of having chemically defined supplements. So the first one is that differences in batch-to-batch composition will lead to lot-to-lot variability, which you want to reduce by using chemically defined reagents. And second, we do see that regulatory expectations for traceability and reproducibility mean that non-CD supplements can add risk to workflows. So to tackle these challenges, we now have the Outgrowth CHO chemically defined for maximum consistency. This is an overview of the different Outgrowth CHO reagents that we have currently on the market. So the Outgrowth CHO and CHO Plus, which Dorothy talked about in her part, they've been on the market for a little longer. We use different formulations, not per se since for the CHO Plus to be better, but the different formulations allow us to accommodate different media types and cell lines. So we typically recommend customers to test both of these and see which of them fits best in supplementing their cells in their workflow with the specific media that they're using. And now recently, the bottom two entries here, we have the Outgrowth CHO CDX and Y, again, two formulas here to make sure that we accommodate different media types. And both of them are available in 5 and 25 ml vials, animal component free and chemically defined. So we've seen that these provide added value for regulatory compliance with the service provider and developer perspective on the right-hand side again, as well as they aim to reduce variability. So we utilize chemically defined products to minimize variability in our workflows, giving consistency in our CHO cell generation campaigns. And we do have some data using different batches. So on the left-hand side, you see Outgrowth CDX, on the right-hand side, CDY. And in our labs, our R&D team has tested, in this case, 3 different batches, and for both, we had very low CVs of 1.6 and 0.6% respectively. On top of that, we've just seen the beta testing data from UJ Biopharma, where we also had very low CVs, and we've seen that the chemically defined will help with regulatory compliance. And compared to a competitor reagent here on the right-hand side, we have market-leading outgrowth, which is superior to some of the competitors on the market. To summarize, before we head to our Q&A session, we've seen that early single-cell conditions are a critical determinant of downstream success, with variability at a cloning persistent through scale-up, banking, and ultimately also regulatory confidence. Optimized chemically defined supplementation can significantly improve single-cell survival, clonal outgrowth, and consistency, which will reduce rework, timelines, and resource burden. And the case study from UJ Biopharma demonstrated how workflow choices, not just clone selection, will drive reproducible performances across cloning campaigns. An integrated end-to-end approach from single cell to stable colony enables more robust, scalable, and regulatory-ready CLD workflows, which will position your team for long-term development success. If you want to learn more about our CHO supplements, please go to the landing page, which is behind the QR code here. You'll find all the data, brochures, and everything that you need, as well as you can request a sample for testing the CHO as created in your lab. We also have the INSI reagents for the hack cells, so if you're working with hack cells, we can also provide you samples for those. With that, I'd like to thank everyone for joining today. I'd like to thank XTalks and UJ Biopharma again for their participation as well, and we will open the Q&A session now.
Well, firstly, I'd like to say thank you very much for that very engaging presentation. I hope everyone enjoyed it. And with that, I'll invite Mohammed and Dorothy to put on their webcams as we join now for the Q&A portion of the webinar. Okay, now remember, you still have the opportunity to send your questions through by using the questions window. So if you're attending GoToWebinar, use the questions window. And for those of you watching on LinkedIn Live events, feel free to post your questions in the comments section. So while you were speaking, I received a bunch of questions. So I don't know how much time we have, so let's try to get through it. So let me start with this one here. This audience member is asking, for teams new to supplementation strategies in CHO, cell line development, what's the simplest place to start? Who would like to take that question?
I can take that.
Thank you.
So maybe, so we started from testing parallel studies, comparing our previous in-house approach that we had against the CHO Plus version. I would recommend you start with that to proof of principle, and yeah, then redefine what is your best working concentrations and conditions. And from that, if you have embedded the supplement or the CHO Plus version into your workflow, you then can implement different CHO Plus versions. For instance, when you want to start with the CHO Plus and also want to test or implement the chemically defined version, you can just then test by A to B comparison with your already established workflow. I think that's something.
Okay, thank you very much. All right, let's jump to the next question here. Would you recommend CHO Prime media as cloning media? Who would like to take that one? See Mohammed smiling. Is that a Mohammed question?
Oh, Mohammed.
Yeah, definitely. Sorry. Yeah. Thank you for the question. So to be honest, yeah, we definitely, I think this is the best would be the recommendation need to be from Dorothy, as she has the hands-on experience regarding using the CHO Prime. So we do definitely recommend CHO Prime as a media and feeds platform. In addition, also in combination with the InstiGrow, CDX and chemically defined, it worked together. I think Dorothea can really agree, and she already showed a very beautiful case, I must say. Did a great job. So yes, simply it is definitely it's recommended from our side. And in order that you would like, if anyone also would like to test this media or this combination, we'd be more than happy to provide from our side I feel free of charge, small amount for the media that could be as a like a testing kit for tests. This we'll be more than happy and glad to offer this. Just write us through the website, or you can also contact us on the info at ugabiopharma.com, and we would be more than glad and pleased to offer you this free of charge testing kit.
Oh, fantastic. Dorothy, anything else to add to Mohammed's answer there?
Yes.
Okay.
I would like to add that our cell line is adapted to the CHO Prime media. This is why also what you have been seeing in the first application note, we tested the commercial available media and we didn't got any clonal outgrowth at all with the commercial available media, and this was why I think was due to the case that our cell line is adapted to our CHO prime media and has therefore the best results. So when you want to work or want to establish the CHO prime media to your cell line, you should keep in mind that you need to take some time for adaptation. This would be something I would like to add.
All right, thank you for that. Okay, let's go to the next question here. I think we have time for a couple more. This audience member is asking, do you see any risk in deviating from the recommended working concentration for Instagrow CHO Plus? I'd like to take that one.
I mean, I can take this as well. So I think you should always stay in a range. I mean, having a recommended working concentration from the manufacturer has reasons, because I think before that the experiments or internal experiments have been done with a certain recommended concentration. However, when you stay in a certain range, I think it can be beneficial. I have been showing in our case that we saw with the original version of Instagrow CHO Plus the best results when increasing up to 2%. However, yeah, I would, do I see any risk? I mean, the product had, or in the supplements, there are also, I guess, some components that could possibly have a solubility limit. So I think this could be a problem when you, have the supplement in a too high concentration that some components are not suitable anymore.
Thank you for that. All right, we'll go to the next question here. What's the average efficiency UGA observes across projects? Another great question. Who's jumping in with that one?
I can do that. Sorry. I remember when I was speaking to the talk, I spoke of good cloning efficiencies, and I maybe have to point out that I'm speaking it from our experience. So I think depending on the cell line you're working with or the conditions that you have in your workflow or in-house, this can be different for anybody, but we are lucky that we define for us 10%, that we see for the most projects that we see around 30% is like good, and everything above is like very, very good. So 30% cloning efficiency means that we can expand 30% of 30%. We got 30% outgrowth from 196 web plate that we seed, which is like really good. And but as I showed, sometimes we got like or get projects where we have like 60%, which is like very, very high, and where
Okay. Yeah. All right. I see I have a couple more minutes, so we can squeeze these two in. You mentioned batch-to-batch differences. This audience member is asking, how was this assessed, and do CLD teams need to do this themselves when adopting Instagrow CHO CD?
Yeah, thank you. I think that was based on one of my slides. So I showed some data from our internal R&D team testing batch-to-batch performance. For these tests, they use 3 independent lots of the CHO CD X and Y, and then for each lot they ran 3 96-volt plates and qualified clonal outgrowth as a percentage of clones reaching a defined confidence threshold. From a customer perspective, we don't believe these studies are necessary as part of a routine adoption. We do very high quality control criteria testing before releasing to ensure consistency between batches, but where required, for example, a very highly controlled development program, we can, of course, provide material from different batches to support internal evaluation as well.
Okay, all right. Well, don't go anywhere. I think there's another question for you now. This audience member is asking, I'm going to squeeze this in. What do you mean by optical clarity of supplements, and how does this influence confidence in clonality?
Sure, yeah, great question. So optical clarity plays a vital role in reducing risk when you're generating or documenting evidence of clonality. Any debris, any particles in your media or your supplements will complicate image interpretation, and that could lead to some ambiguity about the cell origin. So that potentially results in excluding strong candidates from your later stage development. So as a general best practices, you'll see teams filter media and supplements before they do single cell seeding, but with the in situ reagents, this is not required. And this will help to reduce handling steps while maintaining the image quality that's required. So yeah, great question.
Okay, well, that's it for all the questions. Well, thank you very much for answering those questions. We have reached the end of the Q&A portion of the webinar. For those of you interested in hearing more from Novant Biomedical, they have another webinar in May, and when that information becomes available, we will share it with you. Now, if we couldn't attend to your questions, and there was a lot of them out there, please feel free to utilize the email address you see there on your screen, and that's Adam's email address. Okay, thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve our webinars. Additionally, I have shared a link to view the recording of today's event in the chat box, which you can share with your colleagues as well, so they may register for the recording here. Okay? And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to such features as viewing the recording and more. And get even more out of today's presentation by downloading the supporting materials we have available under the Handouts tab, Enhancing Clonal Outgrowth, Optimal Strategies with InstaGrow CHO Plus, and Driving Consistent Single Cell Outgrowth in CHO. So please go ahead and check out those handouts. Now, please join us in thanking our speakers for their time here today. So thank you very much, Naud, Dorothy, and of course, Mohammed. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Bye, Dorothy. Bye, Mohammed. Bye, Naud.